Welcome to another lecture by MedicoMedics, Learning Made Easy. Genetics Chapter 2, Cellular Basis of Genetics. In this lecture, we will present an overview of DNA structure, chromosomes, the process of mitosis and meiosis, errors in cell division, briefly mention ribosomes and protein synthesis, and end with a summary. Now, the basic unit of life is the cell, and it contains the genetic material. We have a nucleus, which is the control center of the cell where the DNA resides. And some organelles involved in genetics include nucleus, which contains the chromosomes, and ribosomes. These read genetic instructions in order to make proteins. Now, DNA is like a twisted ladder with steps made of chemical pairs. And you can see an illustration here of DNA. Now, DNA stands for deoxyribonucleic acid. The shape is a double helix, and its components include a sugar phosphate backbone and nitrogen bases, A, T, C, and G, where A is adenine, T is thymine, C is cytosine, and G is guanine. And these are paired together. So A is paired with T, and C is paired with guanine. And this is what tells your body how to work. The arrangement order. So the function of DNA is it's a blueprint for making proteins. And it's passed from parents to offspring. So we can mention that this sugar phosphate backbone, what it does is that the phosphate group provides structural support by linking to the sugar molecule of the next nucleotide. And deoxyribose sugar is a 5-carbon sugar that acts as the backbone to which the phosphate group and nitrogenous bases are attached. And the nitrogenous base, so these are the variable components of the nucleotides. And these include the 4 types mentioned here: adenine, thymine, cytosine, and guanine. And these bases, right, as we just mentioned, encode the genetic information through specific pairings. Now, chromosomes are thread-like structures made of DNA and proteins found in the nucleus. These organize and store genetic information, ensuring accurate DNA replication and gene expression. Now, humans have 23 pairs of chromosomes, so 46 chromosomes in total. 22 of these pairs are autosomes, so these are non-sex chromosomes, and these generally control our general body traits. We then have 1 pair of sex chromosomes, so these determine biological sex. If it is XX, we have female. If it is XY, then we have male. Now, DNA is tightly coiled around histone proteins, and this forms something called chromatin, which further condenses into chromosomes during cell division. And this compact structure ensures DNA fits within the cell nucleus while allowing controlled gene expression. Now, let's talk about cell division, also known as mitosis. So mitosis is the process by which a cell divides to create 2 identical daughter cells. And this is high yield, 2 identical daughter cells. Why it's important is because we will talk about the process of meiosis. And one way they differ, or it differs from mitosis, is the identical part of it all. But it will make sense as we move along. Now, the phases of mitosis include prophase, metaphase, anaphase, and teleophase. We also have, we may add it here, something called cytokinesis at the end. So in prophase, the chromosomes condense. In the metaphase, these chromosomes align in the center. During anaphase, these chromosomes separate, each to a side. So these so-called centromeres move apart, and half of the chromosomes go to one pole, the other half to the other pole. Then we enter the telophase, and this is where a nuclear membrane forms around these separated chromosomes. So you're beginning to see that it's becoming two. So during telophase, two new nuclei form. And the general purpose of mitosis is to generate growth, repair, and replacement of cells. For example, our skin cells divide through mitosis to heal when we get a cut. So mitosis is how your body makes new cells to grow or replace old ones. Now then, after mitosis, we have another type of cell division, which is meiosis. And meiosis is the cell division that produces gametes, so sperm and egg cells. And it's produced with half the number of chromosomes. The phases include 2 rounds of division. So we have meiosis 1 and we have meiosis 2. And the result of meiosis is 4 genetically unique cells with 23 chromosomes each. The purpose of this is reproduction and genetic diversity. So example, meiosis ensures you get half your DNA from your mom and half from your dad. Now here you can see an illustration where we have meiosis 1 and meiosis 2. So in meiosis 1, which is the reductional division is occurring here, we have homologous chromosomes separating. So in humans, there are 23 pairs of homologous chromosomes, so 46 in total, in a diploid cell. Each pair consists of 1 chromosome from the mother and 1 from the father. Now, during prophase 1, crossing over occurs, where these chromosomes exchange genetic material and introducing genetic diversity. So during prophase 1, and this is high yield, you have the exchange of genetic material and genetic diversity. Now, in metaphase 1 and then following to anaphase 1, this is where they are aligned and then they become separated. So in anaphase 1, homologous chromosomes, not sister chromatids, are pulled apart to opposite poles. This is similar to mitosis. And at the end of meiosis 1, the diploid cell divides into 2 haploid cells, each with 23 chromosomes. Now, these 23 chromosomes are still in their duplicated form, joined at the centromere. Then we have meiosis 2, and this is called the equational division, or where equational division is occurring. So this is where sister chromatids separate. So meiosis 2 is actually very similar to mitosis. In anaphase 2, the sister chromatids of each chromosomes are pulled apart to opposite poles. And the end result of meiosis 2 is that each of the two haploid cells from meiosis 1 divide again, producing in total 4 haploid gametes, so sperm or egg cells in humans, each with 23 single chromosomes. Now, just some key clarifications here. So crossing over happens only in prophase 1 during meiosis 1, not after. And meiosis 2 does not involve chromosome duplication. It simply separates the sister chromatids of the already half chromosome set. So again, the end product, 4 genetically unique haploid cells, each with 23 chromosomes ready for sexual reproduction. So put into a table the difference between mitosis and meiosis. So the purpose of mitosis is growth and repair, and the purpose of meiosis is reproduction. The number of cells in mitosis is 2 identical cells, and in meiosis it's 4 unique cells. So the number of cells is different. Here they are identical in mitosis, and they are unique in meiosis. The chromosome count, same as parent, 46. And in meiosis is half of parent, so 23. So mitosis is essentially for making more of the same cell, while meiosis is for making cells with half the DNA for reproduction. And that's the key difference between mitosis and meiosis. Hope that's clear. Now let's talk about errors in cell division. So we can have nondisjunction, which is a failure of chromosomes to separate properly during meiosis. So it's an error in cell division where chromosomes indeed fail to separate properly during either meiosis 1 or 2. And this results in gametes, so sperm or eggs, with an abnormal number of chromosomes. Now, if you have a meiosis 1 failure, it means that homologous chromosomes do not separate. If we have a meiosis 2 failure, it's that sister chromatids do not separate. And consequences of nondisjunction. So it can lead to abnormal chromosome numbers. For example, Down syndrome, which is known as trisomy 21. It means we have 3 copies of chromosome 21. We can have Turner syndrome, where we are missing 1 X chromosome in females. It's also known as XO. So Turner syndrome, we're missing 1 X chromosome in females. Trisomy 21, or Down syndrome, we have 3 copies of chromosome 21. So an additional copy. We also have another one known as Klinefelter syndrome, which is XX and Y, where we have an extra X chromosome in males. Because males have XY chromosomes, right? So we're talking about a male here. But we have an additional X chromosome. Now, mutations, so changes in the DNA during replication, can be harmless, harmful, or beneficial. Now, the consequences or the impact of these issues is that they can develop developmental delays, physical abnormalities, and even reproductive issues. And yeah. Now let's just briefly mention ribosomes and protein synthesis. So ribosomes are organelles that assemble proteins using instructions from DNA. So the process is DNA transcribes into RNA. And this process is known as transcription. And it transcribes into a messenger RNA. So you will see M and then RNA. And then ribosomes, these organelles, translate this RNA into proteins. And proteins are essential for building and maintaining our body. So you can think of it like ribosomes use RNA instructions like a chef uses recipes to cook meals. And in summary then, DNA is the blueprint of life stored in chromosomes within the nucleus. Mitosis and meiosis are the processes that ensure genetic material is copied and passed on. Errors in cell division can lead to genetic disorders. Ribosomes are essential for turning genetic instructions into proteins. And that's the end of chapter 2. Continue now to chapter 3.